Hello to all of you. I'm going to quickly tell you about this week's labs. So basically, this week you are going to do the streaking lab, and the other document which I posted, it's about the staining and different kinds of stainings. You can just go over it and you can review the questions there. So there's no submission for that. So for the staining, just go over the material I have posted, review the questions, and that's all for this week from staining. Next week we will be actually doing the simple and the differential staining. So basically this week what you're going to do, you are going to do the streaking. Okay, streaking is a very important skill because by streaking we can isolate the colonies. Okay, so basically if you look at this plate, I can explain you from this plate. You can, if you can see here, you can see these colonies. So this is a separate, we call them isolated colonies. Okay, so this is one isolated colony, this is another isolated colony, this is another isolated colony, here is another isolated colony. So these are all different, these are all isolated colonies. So every colony has originated from one single bacterial cell. So basically every colony represents one cell. Okay, so the purpose of streaking is to isolate the colonies, because every colony represents one cell. So which means that if this is one colony, it means this is one bacterial cell. Okay, so basically I will go over the details about this lab with you. I would like to tell you few terms before we move forward. So first of all, what is a pure culture? Pure culture is the one in which microbes are generally genetically identical to one another. So basically pure culture means a culture which has the cells, the bacterial cells which belong to the same strain, which are genetically identical. And mixed culture means when the culture which has two different populations of the bacterial strain. So that is what the mixed culture is. So with the help of this technique called isolation streak technique, or we just simply call it streaking. So basically with this technique, what we do, basically we can have mixed cultures, which means we have the bacterial cells which are of different strains, two different kinds of bacterial cultures we can have. That's called the mixed culture. Suppose there are different kinds of bacteria like E. coli, bacillus. We can have the mixed cultures, and then with the help of the streaking we can have the isolated colonies, which means that with the help of streaking we can have pure cultures, which means if you have a culture which is mixed, you can separate the two type of bacteria. And sometimes we take some samples from different places, different sources, and with the help of the streaking we can actually with the help of the streaking we can have the isolated colonies, and every colony represents one bacterial cell. So with the help of streaking we can actually have the pure culture. We can separate two different kinds of bacterial cells. So that is what is the purpose of this lab. Okay, now let's go over. So you guys are now working with all these plates. So these are, you in the last week in the aseptic technique you have used these kind of plates, right? So this is a solid medium, and on this you can see the morphology of the colonies. So when you will be doing this experiment, you will be actually having two cultures. You are going to mix them. First of all, you're going to make the mixed culture, and then you're going to streak it. I'm going to go over how to do the streaking, and then you will see these colonies. So now the colonies which have originated from different bacterial strains, their morphology is different. Okay, so you can look by your eyes, and when you are looking at the colonies, do not open the plate. You can actually look at the morphology of the colonies. You can look at the color of the colonies, the texture and all that, and by that you can actually differentiate. You can actually differentiate the two different kinds of the bacterial cells or strains. Okay, so so you need inoculating loops, so it should be in your kit. Then you need sterile plastic pipettes, sterile tube, pencil, grease pencil, the paraffin candle. This is your culture tube rack. Okay, for this experiment You will be requiring two different cultures. One is Micrococcus luteus, another is Serratia marcescens. These are the two cultures. If you have, I think it's written in your in this protocol, you can go over it and see. If you have some culture which is not 2 weeks, not more than 2 weeks old, you can still use it. Or I think in the kit they have sent you these two lyophilized bacterial cultures. So they're dry. First of all, you need to reactivate them by adding the broth which has been given to you, and then you are going to take the two cultures and you are going to mix them in this tube. So you are going to prepare a mixed culture. Okay, so these are your cultures, this is your nutrient agar. Okay, so safety instructions are all here. Please make sure you're reading these. I'm not going to go over them in every lab, so you need to be careful. You're wearing these your gloves, you're wearing your glass goggles and the apron. So go for every lab, please go over all these instructions for the safety, because we are working with the microbiology, we are doing microbiology labs and we are working with the bacterial strain. So just be careful with the children and your pets and all that. So please go over this material again every lab and work accordingly. Okay, so basically let's go over the steps of streaking. Okay, and again here are some videos by Carolina. Please watch them. So they explain very well like how to reactivate a lyophilized culture. You have seen in the last lab also, but if you want to watch them again, please watch them. Okay, now I'm going to go over the how to do the streaking. Okay, so I'm going to go over with you each and every step. Okay, mix the contents of each culture tube by tapping or rolling gently between your palms. Okay, using a sterile plastic pipette, transfer 10 drops of Micrococcus luteus into an empty sterile culture tube. Using a different sterile plastic pipette, transfer 1 drop of Serratia marcescens into the sterile culture. Step 2. Okay, so this way what we are doing here, mix the contents of the culture tube by tapping or rolling gently between your palms. Now this is your mixed culture. So you have mixed Micrococcus luteus culture and the Serratia marcescens culture. So you have prepared the mixed culture. So our aim is to separate these two bacterial cultures. So how are we going to do that? Okay, it's simple, it's easy. Okay, obtain a loop of broth from the mixed culture. Okay, and inoculate about 1/4 of the surface of the agar in your plate in a zigzag pattern shown in figure number 1. So you can see here, this is your figure number 1. So you are going to take this inoculating loop and you are going to take a loop full of your mixed culture, and you are going to use 1/4 of this plate and you're going to make a zigzag pattern here. Okay, so you're going to take the loop full. Basically the best thing to do is, you have the culture here in the loop, you're just going to rub the culture on the plate and just spread it and then make the zigzag pattern. Okay, after that what you're going to do? After that place your inoculating loop into the, so once you have made this zigzag pattern, you can discard your inoculating loop. Take a new sterile loop, rotate the plate about 90 degrees. Okay, you need to rotate the plate. So you have made the zigzag pattern, rotate the plate, and then what you're going to do, and pull some inoculum. So take a sterile loop, rotate the plate about 90 degrees and pull some inoculum from the first sector of the agar into the adjacent sector using a zigzag pattern. So this was your first pattern, and then move the plate and now make the, using a new inoculating loop, make this zigzag pattern. Okay, now after that, place the inoculating loop into the disposal jar, rotate the plate again. So again you have to rotate the plate and use a new sterile loop to pull the inoculum from the second sector. So this was your second sector. Now you have to pull the inoculum from here into an adjacent one. So you are going to change your inoculating loop, you are going to rotate the plate, and now you're going to spread the inoculum from this area to this area. Okay, then place the inoculating loop into the disposal jar, rotate the plate and use a new sterile loop to pull the inoculum from the third sector into the final one. So then after that, then after this, this is your third region, and then you're going to take a new loop and you are going to spread it again. That's all what streaking is. Okay, that's all what streaking is. So with this method, what we do, we try to separate the two different kinds of bacteria. Okay, so once it's done, you're going to cover the plate with the lid, and always remember, whenever you incubate your plates, the lid has to be on the bottom, so it is always in an upside down position. Okay, invert the plate so its lid side is down, and then you can, and always remember that, so the lid should be down, and at the bottom of the plate you can do the labeling. And the incubation, so your streak plate may be incubated at 25 degrees Celsius, which is basically room temperature. Okay, and you can check after 48 hours of incubation, you can check your plate. Okay, after approximately 48 hours of incubation, examine your plate with the lid on, and then you can click your pictures. So basically, because your mixed culture has two different kinds of bacteria, you should be able to see with your eyes, okay, with your eyes, two different kinds of bacterial colonies. So there are two different kinds of bacterial colonies, should have different morphology. Okay, so after that, just click the pictures and send me. And for the second the lab material I have posted about the staining, is basically you have to just go over the material this week, and next week we will do the experiment. So that's all for this week. I hope you guys are enjoying the labs, and I know some people have not received the kits yet. We totally understand. So just keep sending me in emails and keep me informed, and I will do my best to let you guys make up your work. Okay, if you have any question, please let me know.